Give it another little bit. We've got about 10 people so far. All right, more people are joining. So let's go ahead and get started. So hello, everyone. So as usual, I'll just kind of go through a summary of the...
Can you put the slides on slide view? We've only got the preview at the moment.
Yeah.
Thank you.
Okay. I'm going to end that. That's better, yeah?
Thank you, yeah.
Try and get this over. Okay. Sorry about that. So as I was saying, typical format, we'll go through a summary of what's gone on since the last meeting, and then really get into the main objective of the meeting is just going through our nomination of M4K 2009 as a development candidate, and then some future work that's going to be happening in the next few months. So we were enrolled in this Tessa Jowell Accelerator program, so I'll describe that. We're going to be collaborating with Matt Dunn at the University of Newcastle in Australia. So he's going to be doing some combination studies with M4K 2009 for us, and then kind of getting into the next steps, which would be CMC work, cell screen and scale up of 2009, as well as some additional ADME and DMPK work. So since the previous meeting, we've really been focused on this RCAS in vivo study being run at the ICR in the UK. There has been some question marks regarding the results of this study recently, and that's really around the bioluminescence of the treated animals. They've had some difficulty with getting the tumors to take, so we'll go into that in a little bit more detail later. However, what I can say is, at least with the data to date, what we see is we're seeing efficacy with 2009 and 2281, but we're not seeing a great deal of efficacy with the M4K 2000 or 2308. We also pulled the trigger on a study looking at the head-to-head metabolite stability and hepatocyte stability between 2281 and 2009. And the results show that M4K 2009 has a much better cross-species stability compared to 2281 and a good overlap of metabolite profile compared to 2281. So really, I think this data suggests that we did see that 2281 had a pretty complex metabolite profile. 2009 does not. So I think this suggests that we have a better rationale moving forward with rats and dogs in a tox study compared to the challenges we might face with 2281. So about 3 weeks ago, we had a steering committee meeting where the group kind of evaluated all the head-to-head data to date between our 3 development candidates and reached a consensus that M4K 2009 should move forward into development. And obviously, it's a huge milestone for the program, and not just that, but I think for open science drug discovery in general. I mean, this is quite rare to progress a program to this point using an open science model. So really congratulations, I guess, to everyone that's been involved in this program right from the beginning. So now I'll just kind of move into you know, the discussions that we had at the steering committee meeting and why we decided to move ahead with 2009. So, you know, as most of you are aware, we spent a lot of time doing a lot of due diligence between these three compounds, and really, you know, focusing in on 2281 and 2308 to determine whether or not they were better candidates than 2009, because they certainly had a lot of really good properties and potential. Not to, you know, very least, we see more potency in the primary assay and in the nanobrett assay with these compounds. All the compounds had good selectivity over other kinases, but really one kind of differentiator is this, even though that we see that increased potency with 2281 and 2308, that didn't really bear out when we test these compounds in DIPG patient-derived cell lines. So there really wasn't any discrimination there. And then we moved into in vivo studies. Again, that increased potency didn't really translate into increased efficacy. So, you know, we looked at, you know, the ADME and DMPK, and I mean, for the most part, all the compounds meet our target product profile, certainly in terms of some of the safety profile, solubility, permeability, you know, everything looked good there. Pretty good microsomal stability as well. However, when we did run this recent hepatocyte stability, you do see that we don't see really stability in any species with 2281, where we see pretty good cross-species hepatocyte stability with 2009. And then I just want to point out one, you know, additional drawback of 2308, which we had discussed previously, was that it is a PGP substrate. And you know, one of the reasons we moved ahead with this was because we do know that it does get into the brain, and coupled with 2281 having good brain penetration, we know that 2281 metabolizes to 2308. So we were hoping that maybe that might compensate for this PGP efflux. But I think you could make the argument that efflux is contributing to the lower efficacy that we're seeing with 2308 in our in vivo models. So when we look at when we dose these compounds orally, again, all three had really good bioavailability, good linear dose response. And we certainly have evidence that each of the compounds cross the blood-brain barrier and get into the brain, which obviously was key for the program. With 2281, we did have this concern about clearance, but again, we were able to explain that high clearance because we did know that 2281 gets metabolized to 2308. Then moving into in vivo efficacy, and you'll all recall that we used this FOP model really as a test for in vivo ALK2 target engagement. And each of the compounds performed, I would say, extraordinarily well in that model. However, when we looked at a patient-derived orthotopic xenograft model, and we've run M4K2009 now multiple times in this model, we certainly see some efficacy. It's modest efficacy, so we do see that 10% extension in survival. However, with M4K2281, that this compound did not perform well in this model, so we did not see any efficacy. So then we decided, like I mentioned, to focus on this our CAS model that's being run by Rebecca and Chris at the ICR. And we ran all three compounds head to head, 50 mg per kg, 5 days on, 2 days off. Now, M4K 2009 was run in this model previously, and we did see, good efficacy and nice extension of survival and decreased tumor burden. Now, when we look at the more recent data with all three compounds head-to-head, and this is data that I presented at that Rebecca presented at the last meeting, looking at the bioluminescence of the tumor-bearing animals. At least with 2308, we didn't see, a strong effect of the compound in terms of the tumor is obviously still progressing. Whereas with 2009 and 2281, we do see that the compounds appear to be having an effect. Now, with the more recent data, and this is what I was alluding to at the beginning, there's been some issues with the model, and it's really been with kind of slow recruitment of tumor-bearing animals that we can use in this study. So what happens is, they have some of the tumors don't take. So we enroll animals based on their bioluminescence, early bioluminescence signal, and then move those into the study. Now, what we did see recently is some of the control animals, kind of the tumor progressed, or the tumor didn't take. So we didn't have a viable tumor in those animals, leading to them surviving, much longer than is typical for this study. So that brought into question, well, are we seeing the same thing in some of the treated animals? And you see, we have some question marks here of some of the animals that, perhaps we're seeing the same effects. So this extension of survival isn't due to drug, it's just due to the tumor not thriving. So, unfortunately, this study is ongoing, the study is still ongoing, but, we'll, in the final analysis, we'll have to drop these questionable animals from the final analysis. But I think what we can say from the data that we have so far is that, we're not seeing much efficacy with 2308. And I would say we're seeing comparable efficacy with 2009 and 2281. But again, we're going to continue on with the study, and once the study is wrapped up, then we can share that final analysis. I'm not sure I know that Rebecca isn't at the meeting today, but does anyone have any kind of questions or comments on this in vivo efficacy data?
I have a quick one, because I don't know how much. I mean, I understand the difficulty here, but at the end of the day, you'll have 5 or about 5, or maybe 5 to 4 mice in each group.
Mm-hmm.
So I mean, I'm not sure the power. I do understand the difficulty here, but I'm not sure if we are going to repeat the whole thing again, or this is it. The efficacy.
Yeah, I mean, we certainly take that offline and discuss it with the folks at ICR. Moving forward, I think we'll certainly look at M4K 2009 and additional in vivo models. But how much we're going to be able to draw from this model, I kind of agree with you. It's it's unfortunate, but these are results that we're dealing with right now. I don't know if Chris is on the line. I don't think so.
Maybe not. Yeah, it's okay. I mean, we can discuss it later. I mean, it is, as you said, you can see the hint here for 209 and 2281.
Yeah, and we have the results from 2009 in the previous studies with this model. So I think we can be confident that the 2009 is showing an effect.
And the vehicle is more or less solid. I mean, this is good thing that you can see the vehicle. I mean, is showing that none of them that has had the tumor got the response, like they are dead or they're not doing very well.
Mhm.
Okay. Yeah, but we can discuss more later.
Yeah.
So sorry, Pete, maybe I missed it, but statistically, you would say these are significant results between 2009 and vehicle. Would you be able to?
Yeah, I think that's a question for Rebecca. But like I said, I think if we go back to the previous studies with 2009, and I think we can have we can certainly have confidence that that we do see efficacy with that compound. Whether or not the results are going to bear out that we're going to see a statistical difference between, say, 2281 and 2009 in this model without adding additional animals to this study, I would say that's questionable.
Yeah, I guess I think it's asking a lot to be able to statistically differentiate the compounds. It's really a yes or no answer. Is the compound you're interested in, based on all of the data that you want to nominate, does that have a statistical significance? And so it's really comparing 2009 with vehicle, I think, is the most important thing.
Yeah, I think what we'd hope for is, would you see some separation between, say, 2281 and 2009? And at least at this point, I don't think we can say that there's any real separation.
Which is okay, because
Yeah,
you have possibly two candidates and you're picking one of them.
That's correct. Yeah, and I think that the other take home is that I think it's quite clear that 2308 just that there is that separation there, that it's not performing as well as the other two.
Right.
Thing too. I mean, one of the things you mentioned earlier is, in the model, a lot of the vehicles should have been killed by now. The tumor take, so you could see the ones that are surviving, those tumors haven't taken. I mean, ideally, all of the vehicles should have been red. Give us a little bit more confidence, but you could see some of the animals, like all the animals, probably the tumor didn't take, so those are surviving.
Yeah. Okay.
Yeah, and that just gets at the statistical significance of vehicle versus
either
2009 or 2281.
Yeah, no, I think your point is valid. Like, getting a statistical significance out of this study. I mean, I'll defer to Rebecca and Chris on that, but I think it's going to be challenging.
And this is not, I mean, that's what happened to us when we had done our first efficacy study with Angle in Spain. Usually all of his efficacy studies, the animals would be dead by a certain day.
And that's what I've seen with this study too.
And for this particular reason, when we did that study, the animals ended up living longer than they typically would. So that really kind of, messed up the data set that we had.
Yeah.
So it looks like it's happening here too.
Yeah, I mean, it just speaks to how challenging these models are.
Yeah.
Okay, so I'll move on just to kind of give you an update on the metabolite ID. So I did show that, we do see good cross-species stability. This is all done in hepatocytes. And then we looked at what metabolites are actually being formed, and can we identify them structurally. So we do see, that good cross-species stability, and we're seeing metabolites that are showing up in human hepatocytes in both dogs and rats across the board. So I think, based on this result, it's certainly much less complex than the metabolite ID that we saw with 2281. So I would suggest this gives good rationale for being able to run a tox study with rats and dogs. So then kind of moving on to the chemistry side of things. So we've made M4K 2009 two different ways to this point, really just varying the starting pyridine compound and switching up the catalysts, but otherwise it progresses kind of in a similar fashion. Now, the real take-home message here is we can make this compound in 3 or 4 steps. So theoretically, this is going to be a much lower cost of goods in manufacturing 2009 compared to either 2281 or 2308, which are 8 or 9 steps. And not to say that those couldn't be made on larger scale, but certainly it's going to cost less, which kind of aligns with our mission of creating a lower cost drug. So really, taken as a whole, I think we have strong rationale to move ahead with 2009, and these are some of the key points that we do see that 2009 is equipotent to the other 2 compounds in cell viability assays, ALK2 mutant and ALK2 wild type DIPG cell lines. We do see better efficacy compared to 2281 in that patient-derived orthotopic DIPG model, and I would say at least at this point, comparable in vivo efficacy in the RCAS model and superior efficacy to 2308. I would say that the metabolite profile of 2009 is a lot cleaner than 2281, and I think we have a better support for using rats and dogs in a toxicology and safety study with this compound. And as I just mentioned, really the not the most important part of this, we wanted to certainly choose the best compound to move forward with, but I think it's certainly good that we have a compound that we can make on large scale at potentially a lower cost for manufacturing compared to the other 2 compounds. So that that kind of summarizes the rationale for moving ahead with 2009, and like I said, I think this is a huge milestone for the team. So does anyone have any additional comments or questions before we kind of move on to the next part, which is what we're going to be now focusing on moving forward?
I'd just say well done for getting to this nomination stage. Great news.
All right. So in terms of next steps, so we were accepted into this called the Brain Tumor Research Novel Therapeutics Accelerator Program, and this is run by the Tessa Jowell Foundation in the UK. And what this gives us is access to an independent committee that can kind of look at our program and give us feedback to help us further de-risk our program moving forward. So what this will give us is about 240 hours of in-kind consulting on our program from an international committee and multidisciplinary committee. So we've basically shared with them all the data that we have on the program to this point, or specifically on M4K 2009, and then highlighted our plans moving forward for CMC and kind of an outline of our tox study, and asked them some specific questions for feedback, mainly around the tox study. So we'll have an in-person meeting with them in November, and Ahmed from the OICR is going to attend this meeting with me in person. He has a fair amount of experience on the safety and toxicology side, so I think he's a really key member to have joined that meeting in person. So we'll get feedback in about 7 or 8 weeks after this meeting. They'll give a full report. So again, I think it's really good that we can take advantage of that, a different set of eyes on our program that can hopefully help us identify any potential gaps that we have in the data to this point, and give us some guidance on what we should be focusing on in terms of a tox study, and what we'll need from our additional toxicology consultants to help us develop a program that's going to be acceptable to the regulatory agencies. So Matt Dunn is a professor at the University of Newcastle in Australia, and I've run into him at a couple of recent meetings. He's very well respected in the field, and I think he's a great addition to have to our collaboration. And his lab is really focused on knowing that this disease is very heterogeneous, so that there isn't going to be a magic bullet for DIPG. So he's really focused on coming up with an optimal combination and optimization for treatment strategies. I think one other important thing to note is, not only is he a researcher, he is a patient advocate, because he runs this other charity called Run DIPG. As Matt's daughter, actually, he was a cancer researcher before, but he has a daughter that actually succumbed to DIPG. So he is a patient advocate as well. So he's really interested in our program and testing M4K 2009, both in vitro and in vivo, and including that in his ongoing ADAPT study. So some of the things that he's done previously, he's been involved in testing ONC201, which most of you know was just recently approved it's the first small molecule therapeutic approved for DIPG. So his lab did a lot of combination work with things like PIK3 kinase inhibitors. So as I mentioned at the beginning, he's a great addition to the collaboration. So really, things are going to be ramping up in the next quarter. The first kind of key thing that we need to do is select a salt form of 2009. So we're going to be initiating a study. We're going to screen probably up to 10 pharmaceutically acceptable acids, looking at things like crystallinity, stability, hydroscopicity, and then from that screen, this can all be done in a high throughput fashion. And we'll take 2 or 3 of those preferred salts, scale them up. We can run a PK study on them. And if we see good pharmacokinetics with that salt, that'll be the salt that we can then move forward with into scale up. We'll be looking at scaling up about 3 kilograms of the material, non-GMP to begin with. Bulk of that material will be used for a GLP tox study, but we'll take a portion of that for formulation development as well. And we'll also be starting analytical method development. So this will be a stability indicating HPLC method, having that developed and validated for use in GMP release and any stability studies that we will be running moving forward. So just to kind of end off, I wanted to highlight again this slide that was prepared by Charles River, and kind of show where we are to date in terms of, we were able to do this cross-species metabolite study and metabolite ID. So we've checked those boxes. And coming up, at least in the short term, this pharmaceutical assessment or SALT screen, as well as the non-GLP scale-up, non-GMP scale-up, sorry. And we're planning some in vivo PK. I'm looking at our doing this in dogs. I guess we should also include a brain to plasma portion of that study. But I kind of want to just open this up to some discussion on what are the next kind of key things that we should focus on in the next couple of months. Do we pull trigger on things like the SIP induction study and phototox study? Does anyone have any comments or questions on kind of these, at least the critical path next steps, which really focuses mainly around the scale up and finishing off some of these additional ADME studies?
I think it's good, Pete. But
Okay.
yeah, I was just thinking aloud. I'm just do you have any backup plan, like any backup molecule or backup project? I mean, I know we have these other two molecules we are thinking of, and we did what you did is rationale why we should go for 209, which is perfect. But then if anything happens throughout this pathway for 209, 2209, what would be the backup plan?
Yeah, I mean, we did kind of discuss this at the steering committee meeting, whether or not 2281 would be a suitable backup. It's certainly more challenging, particularly given I think the metabolite profile. I don't think it's certainly not impossible, but that would be kind of the first thought on a backup. And then is there other things that we can do that may support or exclude 2281 from being a backup? I think on the next tier will be to kind of go through, and this would obviously be a longer term initiative if we choose to do that, would be looking at all of the other compounds that are kind of in the pipeline. There's been a lot of compounds made by Charles River. I just don't have the data package behind them at this point, but that would be kind of the longer term backup strategy if the program was to, if something comes up with 2009 and it does result in us deciding it's a no-go. I think that those are kind of the two backup strategies I think that the program would have. I don't know if anyone else has any other comments or thoughts. I'm sure Metz does.
Usually yeah, I mean, for your backup compound, it depends on what issues you run into with 209 and whether or not, you have other compounds that doesn't have that issue. So your backup could end up, being defined depending on what issues you run into. If you run into an issue 209 that has the same issue in 281, then that's not really helping. So maybe over time, as you go downstream and you run into, if there's any other issues you run into, hopefully there are compounds either from the Charles River effort or from some of the other compounds that we have that doesn't have it.
Yeah, that does. Yeah.
Sorry, I was just wondering, what are your material needs for these studies, and what do you already have material in hand to do?
Yeah, great question. So we have 15 or 16 grams of 2009 on hand. So that's enough to do a salt screen. It would probably take about 5 grams. And that should be enough to do some of these additional ADME studies. I mean, these aren't using up a lot of compound. Potentially a dog PK might use a couple of grams, but we do have, I would say we have enough to do, probably finish off these studies, but it will be critical that we get a new batch of 2009 in soon.
Great.
Okay.
So hopefully the compound synthesis won't be too rate limiting. In other words,
I don't think so. I don't think so. I mean, that getting the large scale 3 kilos would be, but generally these companies are going to do a small batch anyway of 50 grams, which would be plenty for us to do some additional work with.
Okay. Thank you, Pete.
Okay. So in terms of timelines, this was the timeline that we had in the last meeting in July. So we have now chosen that development candidate, but because of some delays, we're going to have to move back a few of these timelines, particularly any of the pre-formulation work, which really isn't critical path, and a little bit of a timeline hit on this scale-up work. We are in the process of getting POs for that, and I didn't mention, but a lot of that those projects are going to be run through the OICR because we will be getting some additional funding, and that funding mechanism works best through the OICR rather than run it through Agora. So we'll be using OICR's procurement process for things like the salt screen and the non-GMP scale up. So at this point, I don't see any, even though we've had to move some of these activities back. At this point, it doesn't really impact the tox study. Although the longer any further delays, then we do risk a timeline hit on the tox study, which will become critical path quite soon. So, I think the timing is good with the Tessa Jowell Foundation, that we're going to get some feedback on the toxicology study, and then we've already kind of spoken to a potential toxicology consulting group that can help us out with that study. But again, we'll need to engage with them and then look at potential vendors and get that lined up for the middle of next year. I think that is all I have for today. If anyone else has any comments or questions or things that they want to bring up. Again, if not, I just want to thank everyone for helping us get to this point. Again, I think it's a really big milestone for the team, and I think we should all be proud that we have advanced the program to this point.
And thank you to you too, Peter, for all that you're doing to move this forward. It's been fantastic to have you on board to shepherd this forward.
Yeah, much appreciated.
Good job, Pete.
Okay, so I think just thinking of the activities in the next month, it's probably not going to be worth having a meeting in a month or so. Probably in 2 months we can reconvene. If there are certainly anything that comes up, I'll share that with the team. But I'm hoping that we can get this initial salt screen started within the next couple of weeks. I think we're kind of moving in that direction, and that study will take probably 6-8 weeks. So we'll start getting some data on that. But we wouldn't have anything in the next 4 weeks. So I'll put something in the calendar for the end of November, I guess.
Sounds good.
Okay.
Thanks, everybody.
All right. Thanks, everyone.
Thanks, Peter. Well done once again.
Thanks, Peter.
All right. We'll talk to you soon.